First, let's talk about endomembrane organelles. The nucleus contains chromosomes. That's the genetic information of any eukaryotic cell. It's enclosed by a nuclear envelope, and it has a double membrane. That double membrane has pores, and these pores regulate entry and exit of materials from that cell, rather from that cell nucleus and nuclear membrane. The nucleus contains a nucleolus. A nucleolus is a very dense region within the nucleus, and that's where ribosomal RNA, perhaps you've heard of rRNA, is synthesized. So rRNA is combined with proteins to form larger and smaller subunits of ribosomes. And these subunits, they exit via that nuclear pore system. They're also assembled into ribosomes, and ribosomes translate messages found on mRNA into the primary structure of what we know as polypeptides. So I said a mouthful, so let's just recap. So in order for DNA to leave the nucleus, it must first be transcribed by RNA polymerase into messenger RNA, known as mRNA. And the mRNA transcript must then be modified before leaving the nucleus to the cytoplasm. In the cytoplasm, the ribosomes decode that information in the mRNA and form continuous chains of amino acids, which linked together ultimately form a protein. This process of DNA to RNA to protein is what we call transcription and translation, central dogma, if you will. And it will be the main concepts that are covered in Unit 6. Ribosomes, they manufacture proteins, and they can actually be free floating in the cytoplasm, or they could be associated with the surface of the endoplasmic reticulum. But they're comprised of ribosomal RNA and proteins. And their main function to synthesize proteins is something that we learn very well in detail, especially come later. But some texts, they do not classify ribosomes as an organelle because they're not bound by any sort of internal membrane. For our purposes, because they are so specialized, we are definitely going to call them an organelle. Ribosomes can be found in two locations. The cytosol, that's where proteins produced here, their generally function is to function within the cytosol like enzymes. We call these also free ribosomes. They're bound to the endoplasmic reticulum or nuclear envelope. These proteins produced here can be secreted from the cell and usually will leave in this transport vesicle system. Speaking of that transport system, it's also part of the endoplasmic reticulum, and that's just a network of membrane sacs and tubes kind of throughout the entire internal part of the cell. It basically functions to not only synthesize membranes, but it compartmentalizes the cell to keep proteins formed in the rough ER kind of separated from those that are free ribosomes. It does have a sorting feature with it. Fascinating thing is that it does look extremely similar in both plant and animal cells. But this network of flattened sac-like structures, what you have to understand is that they're continuous throughout the nuclear membrane, and of course they can be smooth and not having the ribosomes attached to it, or what we call rough because they have the ribosomes attached to it. So yes, it comes in two types, smooth and rough. I always like to show students a visual because actually when you look at this under a microscope, when you can see organelles inside of cell, that's exactly what it looks like and how we can tell the difference. That rough endoplasmic reticulum has these ribosomes that are bound to that membrane, and smooth ones, they don't contain any ribosomes. And they function a little differently too, because the smooth endoplasmic reticulum synthesizes lipids. It also breaks down to metabolized carbohydrates, which essentially detoxify the cell. So its function is different than the rough ER. The Golgi apparatus is my favorite organelle. It contains these flattened membranous sacs called cisternae. These cisternae are separate from the sacs that are in the cytosol, and each of them is actually not directly connected, regardless of how the picture makes it look. It actually has directionality. If it's a cis phase, it's receiving vesicles from the endoplasmic reticulum. And if it's what we call trans phase, it's sending vesicles back out to the cytosol. So the Golgi complex functions to receive transport vesicles with materials or coming with materials that are from the endoplasmic reticulum. And then it modifies these materials. It ensures these newly formed proteins are folded correctly, kind of like a double check process. And that if they're not folded correctly, it modifies them to put them back in the right shape. It sorts those materials. A lot of times it'll add additional molecular tags to those proteins, and it packages them and puts them into a new transport vesicle, and that will eventually exit the membrane. We call that exocytosis. Lysosomes are an internal sac that hold hydrolytic enzymes, and the function of those enzymes inside of the lysosome is to hydrolyze macromolecules, and this takes place within the animal cells. Lysosomes can also recycle their own organic materials. That's called autophagy, where it can essentially renew its own internal materials. Another organelle, peroxisomes, they're very similar to lysosomes, but the membrane-bound metabolitic compartment does something different. It actually works as a catalyst. It catalyzes reactions that produce H2O2, which is peroxide. So enzymes in the peroxisomes, they break down things with H2O2, and they break it down from that peroxide to water. There's lots of vacuoles inside of cells, but let's talk about a few just real specific ones. So the large vesicle that stems out of the endoplasmic reticulum and the Golgi apparatus is committing this selective transport. It can work in 3 different major ways. It can work as a food vacuole, it can work as a contractile vacuole, and it can also work as a central vacuole. The central vacuole is only found in plants. It stores water, ions, other inorganic components, and that's important when it comes to turgor pressure inside of plants. A contractile vacuole is going to maintain water levels inside of all cells. And a food vacuole does kind of what it says. It actually commits phagocytosis. We call that cell eating. And that is when it uses those digestive enzymes that are inside lysosomes to commit phagocytosis.